Welcome to the Chapter 11, Section 2 podcast. This is on probability and Punnett squares, and we're going to learn a few more terms about heredity that we're going to use. So, how did geneticists use the principles of probability? Mendel was one of the first people to use the principles of probability to explain the results. He actually did a great job using probability. What is probability? It is the likelihood that a particular event will occur, and you can use probability to talk about a coin flip. The coin flip will be, there's a 50% probability that it will turn up heads. You can talk about the probability of rain for the day. There's a 20% chance of rain today. So probability is used, you've heard probability before and you've seen it used in a lot of places. So the principle of probability can be used to predict the outcomes of genetic crosses. So how do geneticists do this? They use a diagram known as a Punnett square. And from this diagram, you can predict and compare the genetic variations that will result from a cross. And here's a picture of a Punnett square. We need to talk about a few things. A capital letter represents a dominant allele for a trait. So right here you can see here's a capital letter here, here's a capital letter here, here's a capital letter here. These are all dominant alleles. And a lowercase letter represents recessive allele. And here's a lowercase letter right here. Lowercase letter represents a recessive allele. So in this example, we're talking about the characteristics of tall and short. Big T will stand for tall, little t will stand for short. Often they'll use a letter that represents the dominant trait. In this case, obviously, big T for tall. They wouldn't use an S. Because it S is the recessive trait. So we have two parents right here, one right here and one right here. And they're going to make gametes. And the gametes, remember, are only going to have one of the two alleles. So this organism right here is going to make these two gametes, and this organism here is going to make these two gametes. And in this case, they happen to be the same type of organism where it has one big T and one little t for its two alleles. So the possible gene combinations for the offspring appear in the four boxes. So here we go. We got this T here and this T here, and they lead to this double T or big T here. Let's look at this one right here. We have a big T here, a little t here. So if you follow it over, we have a big T, little t right there. And that's how you do it. And you do it for each of the squares that you have in each of the four boxes. So here's one of those terms we need to understand. Homozygous. So if we have an organism that has two dominant alleles, say for instance case, it would be homozygous. We could also have an organism with two small alleles. Let's jump back for a second. This one here is also homozygous. It's got two little t, little t. It's little t, little t. It's homozygous. So if they have two identical alleles for a particular trait, we say that that organism is homozygous. And if they have two different alleles for the same trait, we call them heterozygous. And here's the heterozygous plant right here. It has a big T and a little t. So homozygous plants are what we call true breeding. We talked about this at the very beginning of Mendel's experiments. He got a bunch of true breeding pea plants, and they were homozygous for the trait that he was interested in. And heterozygous organisms, we say, are hybrids. We said that hybrid is a result of the cross of two organisms, purebred, that have the opposite forms of the trait. So these are hybrids. So heterozygous is a word hybrid is a word we often use for heterozygous. Now, those are the this is the phenotype now we're talking about, which is the physical characteristics of the plant. And if you look, both of these plants are tall. So the tall plants have the same phenotype, the same physical characters, but they may not have the same genotype or genetic makeup, which means we go back to here. This one's big T, little t. This one's big T, little t, big T, little, big T, and big T, little t. They have the same phenotype. They're both tall, but they have a different genotype, different genetic makeup, because they carry different alleles. So in the case of our cross that we did before, and we'll see it again in a Punnett square in a moment. 1/3 of the plants are big T, little t, while 2/3 of the plants are big T, are big T, big T. 2/3 of the plants are big T, little t. So these plants have different genotypes, but the same phenotype. So let's look at that Punnett square again, now that we know what phenotype and genotype are. If you look, we have 1/4 of the F2 plants have 2 alleles for tallness right here. 2/4 or 1/2 have one allele for tall and 1 allele for short. 1 allele for tall, 1 allele for short. But you look, these three are all tall plants. There's this genotype, and then there's the big T, little t genotype. And then 1/4 are going to have the 2 alleles for a short plant, little t, little t. So because the allele for tallness is dominant, like we just said, 3/4 of the plants will be tall. These three right here, 3 out of 4. So the ratio of tall plants to short plants is going to be 3 to 1 in this cross. So what Mendel found was that when he did these experiments, and he did them with hundreds of pea plants, he kept finding 3 to 1 ratios in this situation where he crossed a hybrid tall with another hybrid tall, heterozygous tall with another heterozygous tall. He ended up with 3 tall plants to 1 short plant. So let's talk about sum this up on probabilities. Probabilities predict the average outcome of a large number of events. That means if we do 400 crosses of 2 heterozygous plants, we're going to end up with a 3 to 1 ratio. So we should have roughly 300 to 100 ratio. However, it cannot predict the precise outcome of any individual event. Meaning, if we took one seed and looked at the Punnett square and said, oh, this is going to be tall or this is going to be short, we can't do that. We can look at a large number of offspring and use the prediction. And also, as we get more and more offspring, we'll see that that number comes closer and closer to 3 to 1 ratio in this case. A good example is a coin flip. If you took a coin and you flipped it, say 5 times, you might get 4 heads and 1 tail. But if you flip a coin 50 times, a larger number, then you're going to get very close to 25 heads and 25 tails. And that's what we're talking about when we talk about this last point.